Bismillahirrahmanirrahim and welcome back to this next video, and this is the 10th video of the chapter 2 of Immunology. From the last couple of videos, I'm telling you about the neutrophils, how the neutrophils they are formed, and what are the important contents present in the neutrophils. And I've told you that there are special granules in the neutrophils, and we usually classify them into four classes, and we were focusing on the primary granules, also known as the Azurophilic granules, from the last couple of videos. Now in this particular video I want to focus on another type of the granules that are present in the neutrophils, which are known as the secondary granules. Now, if you remember, I've told you in my previous videos that during the process of the granulopoiesis, the granules they are made when the neutrophils they move from one stage to another stage. And when you talk about these secondary granules, they are usually made when the neutrophils they move from the myelocyte stage to the metamyelocyte stage. We discussed this image in a great detail in my last videos, and I'll share the link in the description. So when you talk about the secondary granules, they are usually made when the neutrophils they move from the myelocyte to the metamyelocytes. Now, what is present in these secondary granules? If you look at the composition of the secondary granules, it contains a lot of antimicrobial component, and in this particular video I want to focus on the lactoferrin. Now this lactoferrin, if you talk about this, this is an iron binding glycoprotein. I hope you know the concept of the glycoprotein. After the post-translational modifications, the proteins they are changed in a variety of ways. And one of the way in which you can change the protein is by adding the carbohydrate moieties. And when you add the carbohydrate moieties, we usually call them as the glycoproteins. So when you talk about this lactoferrin, this is actually a glycoprotein, and it has the potential to bind to iron. Therefore, we call them as the iron binding glycoprotein. And this lactoferrin, it belongs to the family of the transferrin. If you talk about the molecular weight of lactoferrin, it is 78 kilodalton, and it is found in a variety of the body fluids. For example, the milk, the saliva, tears, mucus secretion, as well as in the neutrophil secondary granules. So we are discussing the lactoferrin, which are present in the secondary granules of the neutrophils. One thing to keep in mind, these secondary granules, sometimes they are also known as the specific granules. So the specific granules of the neutrophils, the secondary granules of the neutrophils, they refer to one in the same thing. These lactoferrin, what they can do is they can sequester iron. And when you talk about the function of the lactoferrin in the immune system, so when they sequester iron, this iron is an essential nutrient for the microbial growth. So if the lactoferrin sequester iron, the microorganism they will not be able to get the iron, thereby inhibiting the growth of the bacteria or the fungi, whatever the pathogenic organism is. Now, why this iron is so important for these bacteria, and once you for these bacteria or fungi, and once you sequester the iron by using the lactoferrin, how it is going to badly affect the microorganism. If you talk about the functions of the iron in these pathogens, the first important thing is they are very important for the energy metabolism. So iron is a vital component of many enzymes in the pathogenic bacteria or fungi. In these enzymes, they are involved in the cellular respiration and energy metabolism. For example, if you talk about the cytochromes, they are very important iron containing proteins. And these cytochrome, they facilitate the electron transport and energy generation in the form of the ATP in the bacteria, fungi, or other pathogenic organisms. So if you are going to sequester the iron, that means that these cytochromes, they are not going to work. They will not be able to generate the ATP, there will be no energy available for the growth of the microorganism. Thereby you can stop the infection of that particular pathogen. Another important you can say function of this iron in these pathogen is that they are very important for the DNA synthesis. For example, there is an enzyme which is known as the ribonucleotide reductase, and this ribonucleotide reductase is responsible for the conversion of the ribonucleotides to the 2' deoxyribonucleotides, and thereby provide the precursors needed for the synthesis of the DNA. So if you sequester the iron, that means you are making the ribonucleotide reductase non-functional. The bacteria or other pathogens, they will not be able to synthesize their DNA. That means they cannot go for the growth or for the cell division. Another important function of the iron, or another important you can say component where the iron is important, they are the virulence factors. Now, many pathogenic microorganisms, they express iron-regulated virulence factors, and they are known as the siderophores. And the siderophores, they are actually molecules that scavenge iron from the host to support the microbial growth and in the establishment of the infection. Let me give you some of the important examples of these iron-regulated virulence factors. If you talk about the E. coli, the E. coli is going to produce this enterobactin, and this enterobactin is actually a siderophore. That means with the help of the enterobactin, the E. coli can actually take the iron from the host, and it can use it for the microbial growth and for the establishment of the infection. Another important example, the salmonella, very important foodborne pathogen. They are going to produce the enterobactin and the salmonellin. The Mycobacterium tuberculosis, which is the causative agent of the tuberculosis, that is going to produce this mycobactin. If you talk about the fungi, for example, the Aspergillus species, they are going to produce specific siderophores. You can call them as the ferricrosin and the ferrichrome. And if you talk about these one, like the E. coli, the salmonella, the Mycobacterium tuberculosis, or the Aspergillus, by producing these siderophores, they can scavenge the iron from the host, and they can support their growth, and that can help them in the establishment of the infection. Now, what the lactoferrin do, that this lactoferrin is actually it can possess 2 high affinity iron binding site, each capable of binding to the ferric iron. What this means is that the lactoferrin that is going to bind the iron, the siderophore, they are going to bind to the iron. So when the lactoferrin, they bind to the iron with a very high affinity, that means that these siderophore, they will not be able to extract the iron from the host. Therefore, their growth will be stopped. The establishment of the infection, that will be stopped. And the binding capacity of the lactoferrin to iron is particularly strong, allowing it to effectively capture and sequester the iron ions. Now, as I've told you that these pathogenic bacteria, they produce these siderophores. These siderophores are actually low molecular weight chelating agent. In this particular case, they are chelating the iron, and they bind to iron and facilitate its uptake. Now, these lactoferrin and the siderophores, they are going to compete for the iron. The lactoferrin bind with the iron with a high affinity, a strong interaction with the iron. Therefore, when the lactoferrin, they bind the iron, the siderophore, they are usually not able to bind the iron. Then they are not going to extract the iron from the host. Therefore, their growth and the establishment of the infection, that will be stopped. Therefore, this lactoferrin, that is part of the host defense mechanism. So this is one of the important antimicrobial component that you see in the secondary granules or the specific granules. In my next video, I'll talk about the other important antimicrobial component present in the secondary granules. So if you like the video, please subscribe to my channel, hit the notification bell, do share it with your friends, and if you have any question, you can ask me in the comment section. I'll see you in the next video.